Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Went, and I'm the founder of Everything Epigenetics. Today, my guest is Dr. Candace Lewis. We have a very fun podcast for you all today. We really talk about Candace in the lab she runs called BEAR. It stands for Brain Epigenetics and Altered State Research at Arizona State University. We really dive into what she studies and talk about how this affects every aspect of life. She not only looks at everything at the cellular level, but she also looks at everything at the physiological level and then the family level and the societal level as well. We talk about her editorial she wrote in 2023 called "What is Up with Psychedelics Anyways?" We know that's a new, sexy, hot topic nowadays. We also talk about how epigenetics sheds light on psychedelic therapy, and we even break down one of her pilot studies published in Frontiers in 2023 that suggest DNA methylation of the glucocorticoid receptor gene, NR3C1, is associated with MDMA-assisted therapy treatment response for severe PTSD. We also talk about her psychedelic genome project, why it's important, and how you can get involved. And we really also discuss during the entirety of this call just all of the projects that she's working on and what really excites her. We then end by talking about her research focusing more on social stress exposures and how those experiences can shape dynamic regulation of gene systems underlying cognitive, emotional, and behavioral health through development. A little bit about my guest today. Dr. Lewis grew up in rural Alaska, where she was exposed to high rates of poverty, mental illness, violence, and addictions. And those personal experiences that she went through really drove her commitment to revolutionize mental health understanding, treatment, and even policy change. To this end, she earned a master's in counseling, a PhD in behavioral neuroscience, and studied genomic science at a leading institution and studied psychedelic science at the University of Zurich. Like I mentioned, she's at the BEAR lab, where she really focuses on three different things: the impact of early life social experiences on epigenetic regulation of gene systems involved in mental health. She even studies the relationship between peripheral epigenetics and brain structure, so the function, the microbiome, composition, and even behavior. And then number three, which is my personal favorite, is we dive into on the podcast as well, is the potential of psychedelic assisted therapy to reduce symptoms through psychological healing and epigenetic alterations. Through her research, she aims to acknowledge the harm caused by psychology and genetic sciences on minority groups, increasing diversity in training and study cohorts, and really pushing to change policy to improve health for all. When not working, Dr. Lewis enjoys family time with her three sons and wife, going to festivals, hiking, running, reading, creative writing, and talking all things spirituality, social justice, and science. Now for my guest, Candace Lewis. I hope you all enjoy. Welcome to the Everything Epigenetics podcast, Dr. Lewis. I'm so excited to have you here today. Thanks for being here.
Of course. I'm happy to be here.
Yeah, so we're just going to jump right into it. You run the Brain Epigenetics and Altered States Research Lab, or BEAR Lab, at Arizona State University. And this is the first time we've ever chatted. I know I reached out to you via email to get you on here. So I know just very little about your story and how you got into this field besides what's on your beautiful lab website. So kick us off and tell me more about yourself, how you ended up in the BEAR Lab. Tell me a little bit more on what that journey looks like.
Wow, so much, so many years. Okay, so I did not grow up thinking I was going to be an academic or a scientist. I thought, which a lot of kids in the 90s did, I don't know if they still do, that scientists were like old white dudes wearing lab coats playing with green fluids. And it was in undergrad that I realized that I really loved school. And I just was working really hard. I was working full time while getting straight A's. And I happened to come across this fellowship called the Truman Fellowship, which I decided to apply for, not really knowing what it was. And it turns out that it is a highly competitive national scholarship to go to grad school. After an arduous application process, I got it. And it's so funny because during the interview process, they said, "Do you want to get a master's or a PhD?" And I was like, "I don't know what the difference is," because that's how much I was not prepped to go to graduate school. And it was in graduate school that I really kind of was exposed to the idea of studying stress and the idea of studying early life stress and the idea that there's this whole science around understanding how it affects us at the biological level. We've known for a long time that behaviors are associated with early life stress, right? It's a really large predictor for most psychiatric disorders, PTSD, depression, anxiety disorders, substance use disorders. And that really called to me because I grew up in a chaotic home where I was in and out of foster care. And we were also, we lived in deep poverty and we were in rural Alaska. So I saw a lot of this around me in a lot of different ways. I mean, Alaska is has some of the highest rates per capita for a lot of these disorders. So I was in a lab, I was studying stress. I was like, find myself now, and I'm an academic, I'm a PhD. And it was my first year as a grad student that I learned about epigenetics. And it was just kind of just coming onto the scene, it was just getting really big. The field was exploding with the idea that epigenetics are not just static regulators of cellular differentiation, but that at least some parts of the epigenome, it can be responsive to early life experiences. And by responsive, I mean that it's going to change, it's going to adapt, and that those regulatory changes stay consistent or are long term. And in effect, they change gene transcription, which changes everything about the cellular makeup of the brain, which then the brain is what's regulating all physiology and behavior. So I was just like, this is amazing. My mind was blown. I fell in love immediately. And I'm like, this is it. And at the time I was studying addiction, an animal model of addiction. And I was like, so early life stress influences drug intake behavior in rodents. We know that. So instead of staying in this cortisol, stress reactivity, depression findings that the field was really focused on, I decided to look at, so is early life stress influencing epigenetic regulation in brain regions associated with addiction? And so that was my graduate work, and it was great. We really showed this causal pathway between early life stress altering epigenetic regulation in the nucleus accumbens core, and that being associated with the animal's different methamphetamine intake behavior. We were also able to reverse those epigenetic changes and showed a decrease in drug intake behavior. So that was my background. That's what started it all, and I've been doing it ever since.
Yeah, thanks for the introduction. That's quite a story. The application of really how understanding how stress affects our body is something I'm personally super passionate about as well. I'm just a naturally stressed out person, super anxious all of the time, and really understanding what I can do to actually reverse that. So I want to ask you one more question before we move on. You mentioned that in the mice you were, measuring the epigenetic patterns, and you actually saw a reversal in that stress in those patterns too. So what caused that reversal?
Well, unfortunately, it was a research method, not anything that we can apply to ourselves in life. It was it
Got it.
was a viral vector injection. Yeah.
Got it.
Into the
Okay.
brain region. But I
Perfect.
want to say though, because I think it's so important, right? Because I find myself in these types of conversations because I study how, maternal care and all these things, and all the moms in the audience, and I'm a mom too, so I get it. They're just like, oh my God, I'm messing up my kid. I'm messing up their epigenetics. And I'm like, no, 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 no. I really think that it would behoove us to understand this new, incredibly complex, beautiful thing about our brains, that it's molecularly adapting to our environment, that it's a good thing. So the way that I like to kind of explain that to people is, if you're born human or primate or rat, you're a pup, you're developing, and you're born into an environment that has low maternal care, low resources, high stress. It would make sense, and it is highly adaptive for your brain to be like, okay, this is what I got, and I need to be hypervigilant. And it modulates gene expression for you to be able to fight for and seek your needs. And hypervigilant is another word that we call anxiety today.
Gotcha. Yeah. So
Um,
yeah, go ahead. Keep going.
I just think that, I think that sometimes it's important to see that we have, our current Western model of mental health. We have these disorders, and then we all think about them a lot. People say all the time, oh, I have OCD. Well, they don't really have a diagnosis of OCD, but they see themselves in that description. And a lot of times people get caught up in, what does that mean about my brain? What does that mean about my genetics? And what does that mean about my children? And I think it's just so important to always bring it back, even though I study the biology, that the disorder is just a list of behaviors. It's a list of behaviors that you self-reported at a time. And maybe at that time, they made a lot of sense. You're not biologically, stuck here to a
Got it.
certain degree, right? It's all spectrumy.
Yeah. So yeah, you're saying, you can ebb and flow in and out of kind of these, I don't know if you want to call them then, diagnoses
I
or,
know, yeah.
time of how you were feeling, right? And in terms of those outcomes, because yeah, I mean, I can even reflect back on myself and say, hey, I felt more anxious, during this time because of these things or, my environment, my behavior, my surroundings. And so I think what I'm interpreting from what you're saying, Candace, is more, you're in control, right? It's how you're feeling, and different things can trigger that, again, that environment or that behavior. And maybe it's not necessarily always a negative feeling too. There's maybe some positive connotation behind that.
Yeah, I mean, I think control is a tricky word to use,
Hmm.
especially when we start bringing in systemic issues. However, I guess, yes, the idea is sometimes anxiety makes a lot of sense for what life's throwing at us. And sometimes being really sad makes a lot of sense for what life's throwing at us.
Yeah.
And yeah, and I'm just really interested in that throughout the lifespan, right? Our brain is adapting to that.
Yeah, I like that. I'm gonna have to sit on that more and like
Mhm.
actually, do a lot of like self-reflection. So thank you. That's a really good, introduction to this entire podcast. I want to dig a little bit more deeper though into exactly what you're studying. So yeah, if you want to give your own explanation of, what is the Bayer Lab, which is an awesome acronym, and what exactly are you studying now? What are you most passionate about?
So I think about my research in two kind of parallel lines. I like to look at the things that are affecting us early in life that maybe predisposes towards these behaviors that we call psychiatric symptoms. So, okay, so that's the one track. What are the things that are happening to us that are potentially altering epigenetics of genes that are involved in things like mood and cognition and stress physiology, right? And these are the underlying pillars of the psychiatric disorders. Additionally, I'm interested in if we're so susceptible to these hard experiences, to these experiences that are telling us you're not that safe, then what's the inverse? What are the experiences that we can be having that say like you are safe? And does our epigenome respond to those as well? So in that sense, I also am interested in looking at psychedelic therapies and the idea that potentially, in a very similar way in which our body responds to these moments that are screaming like you're not safe, so you should be responding and altering. Is it possible that different psychedelic compounds are inducing a similar type state where your body is responding and being like, okay, I actually am safe. I should be modifying to adapt to that newfound experience.
Mhm.
So, but one thing I want to unpack about that quickly is because the semantics, right? We'll always get back to the semantics. So, trauma. People ask me this a lot. Well, what do you think trauma is?
Mhm.
And I think that, we have the ACE lists, right? Those 10 things are like the yes or no, and the DSM is still pretty hardcore clinging to the idea that there has to be like a threat to safety, whether perceived or even witnessed of someone else. However, I like there's so much data showing that it doesn't have to be this really, really strong, what we would call trauma to shape us. So like what I mean by that, you have a lot of people, they go to therapy and they're depressed and maybe they're anxious and maybe they have a substance use issue. And they're like, nothing happened to me. Nothing happened to me. I had a great childhood. My parents were married. We had resources. I was on the basketball team. And so it's confusing for them because they can't check the trauma list.
Mhm.
However, I think that there's so much more going on with like attachment and safety. And if you look through the attachment field and you look at the like long-term associations with disorganized or non-safe attachment while you're younger, they look a lot like the same effects of trauma.
Gotcha. And
And
you're just
so,
by that too, you're just talking about like different attachment styles. Is that correct? Just to clarify.
well, that one I think is really tricky because like there's the science of attachment styles, and then there's this like fake pseudo parenting theory that came out a long time ago. It's like never put your baby down. So that word gets really tricky to use to people too. I'm using the word attachment because I want it to encompass so much more of a childhood experiences than just trauma. But it's, I think that probably there's a lot more layers to it. So like emotional availability is something that comes up, which, could be nested under attachment. But really it's the perception. It's the childhood perception. Am I loved? Am I safe? What are the conditions that I have to meet to earn those things? And then I think how far that child has to deviate from their authentic self to meet those conditions is a very it's a experience that is very shaping throughout childhood. So I think what kind of ends up happening with a lot of these people who can't check the trauma box is you dig deeper and you're like, okay, yeah, maybe I really felt that mom and dad didn't love me unless I got all A's.
Mm-hmm.
So no, I'm not talking about trauma right at that moment, right? We're not calling it trauma, but I'm interested in the whole gamut because there's so much literature out there showing that those experiences shape us too. Those experiences can
Yeah.
underlie anxiety.
Yeah.
Yeah, a lot of high performers. This, right now, I think we are seeing a world of like everyone in a certain age bracket. It's like, I'm so anxious. And like, I really think that there's this like generational reason of, the
Yeah.
expectations that were put on them to school, work, sports, manners.
Absolutely. I think I think I fall in that category. Absolutely. And it's interesting too. So yeah, you're doing a lot of cool stuff at the Bear Lab. You're interested in what's like happening to us to affect these disorders, the experiences we're having, showing us that if we're safe or not. And we're really going to dig into that on the podcast today. But I like that you also mentioned, hey, it doesn't have to be trauma. It can be this like attachment. It can be trying to fulfill this image. Or I really like how you put even deviating from your authentic self. So
Mhm.
I'm sure, yeah, trauma, you can talk about it in many different ways or other things that have happened to you. And I don't know that I've even ever shared this on my podcast before either, but, grandmother passed away from Alzheimer's. That was a really large event that happened in my family, and not necessarily even myself going through it, but seeing like my mom go through it, right? And seeing how she grieved. You never want to see people you love being upset and hurt. My dad passed away at a super young age, unexpectedly from a cardiac related event, right? So those I would say, maybe they fit in that traumatic kind of category, but also I did just feel the need to perform. Like I got straight A's. I played, 5 sports year round, and not because my parents were like, you need to get straight A's, you need to get straight A's. But I did feel this like untalked about like pressure, right? Whether it was like definitively stated or not. And that could be the underlying reason. I am obviously, gonna dig into that a little bit deeper as well. But I thought it was an interesting piece.
Thank you for sharing all of that, right? And I think that your example, it so shines a light on why this is hard. It's not a list of 10 things we can check yes to. Like you
Right.
said, maybe it was never even said to you, but it was felt.
Right. Right.
Yeah.
And it just goes kind of unsaid. And yeah, you're trying to meet these expectations. And it's not necessarily the truth, but rather the story you're making up about it. So
Yeah.
yeah, I think it's just cool. You're looking at all different aspects, not just like the ACE, kind of checkbox or scores. You're really digging into almost like that social aspect of like
Mhm.
the humans and us like actually living, which I think goes missed a lot of the times in this type of
Mhm.
research.
I constantly am reminding people when I talk about my research that like for the most part, I'm never talking about an individual. I'm talking kind of about a system and systemic issues and cultural, right? Because so here, let me give you an example. I have a paper that looks at harsh parenting. So parents self-reported, and it happened to be all moms. We used primary caregiver, and that cohort it was all moms. They self-reported when the kids were 2 years old, and it was things like, screaming, yelling, threatening, throwing, spanking, the type of things that you cannot get your children removed for DF from child services. And then we looked at the children's epigenetics when they were 8 years old. Parents' self-report of harsh parenting was able to predict DNA methylation of NR3C1, the glucocorticoid receptor, and other HPA genes. So for a little bit of background to explain that system. So the HPA system is the hypothalamic pituitary adrenal system. It is the system that is responsible for your cortisol response to stress. And chronic levels of chronic high levels of cortisol has been well documented to be associated with many psychiatric and neurodegenerative disorders, actually. And it wreaks havoc on your immune system. Like, this is a well-known thing. Chronic cortisol is not a good thing. And the Weaver '04, the big paper that made the huge splash that looked at maternal care in rodents, this was the gene that they really looked at. So we were, and then after that, a lot of the human studies followed up with these big trauma cohorts. So, the more than the trauma category cohorts. And I wanted to follow that up with harsh parenting. And we found that same relationship, right?
Yeah.
And so I'm writing the discussion of this paper, and I stop and I think, oh my gosh, I sound like Freud. I was like, I can't publish this. I'm saying everything's mom's fault. Like, that's kind of like the take home of the message. Moms, no matter what you do, you're going to screw your kids up, and stop yelling. It's like, I yell at my kids sometimes because life's hard.
Oh
And
yeah.
sometimes it happens. You know what I mean? So I was like, I can't put this out there. But I think it's so important. So what do I do? So what I did is I dove into the literature of what predicts harsh parenting. It's a societal issue. It's not mom's fault. Okay. So I was able to find all this research. So, like, here are some of the high hitters: lack of sexual education, lack of birth control availability, poverty, marriages, forced marriages, not chosen marriages, unequal distribution of labor, not having paid paternal leave. These are systemic things that are all throughout America, and it leads to very high levels of stress for millions of us that are living paycheck to paycheck, did not have access to sexual education, yada, yada, yada. And so I put that paragraph in there. That is a very rare paragraph to find in a molecular biology research paper. But I think it is so important.
Yeah.
Last thing, I was giving that talk one time at an international conference, and I stopped in the middle of that slide and I looked around the room and I go, none of you understand what I'm talking about, because none of them were Americans. They were all they were all from a Western European country, and all of their countries, all of those policy things that I just listed, they don't share those issues. And I was just blown away by, I study the family, I study the individual, and I study molecules, and I'm a neuroscientist, so I'm all about the biology. But I think for me and for my lab, it's so important to recognize how the bigger picture is affecting all of our biology.
Absolutely. Well said. Very well said. You're looking at the entire picture. I literally got like chills and goosebumps as you were going through that. I'm like, wow. Yeah, so that actually was not a paper I was like going to bring up pretty intentionally on the agenda today, because I like got the paddle. Like, I remember like my mom and dad, like, screaming, yelling. Like, I got spanked when I like would get out of line, and I have 2 younger sisters, so it got better. Like, they didn't get, as much punishment as I did growing up. I'm the oldest. And I was like, you know what? I don't think I'm going to bring that one up or ask about it, because like I'm kind of embarrassed of like, that like entire thing. And I don't want people to think my mom's a bad person, because she's
Mhm.
not, you know what I mean? But yeah, I must have even skipped through and obviously, didn't dive into all of the details there and like hit on that like one paragraph, because that is so important, where you're like, okay, I'm not going to blame the person for being that way. I'm going to ask like, why? Like, why are
Mhm.
they actually being that way?
Mhm.
So that's huge. Thank you for
Yeah.
explaining that.
And like, thanks for sharing. And no, your mom's a wonderful mom. And it's even so in harsh parenting and corporal punishment, it is a very tricky topic right now, right? It's very tricky. And what we're finding is that these kind of like negative long-term consequences, it's mostly in white kids, and people are having a hard time replicating it in African American and Hispanic cohorts. And I think that one could, go on for a long time to kind of say why. But to me, they culturally, they also have much higher levels of emotional availability with their children.
Mm.
So kind of back to your experience and like, witnessing death in your family. So hard, right? It was so hard. And getting spanked by your parents, it could be so hard. But all of these things that children can be exposed to, the really important other side of that, it's not just the exposure. It's what's the package.
Yeah, everything together.
Everything together. What are the resilience factors? Because if I was not close to my parents at all, um, and I feel like they didn't know me, and I was kind of scared of them, and I got spanked. Like, I could see how that would be really horrible, as opposed to if I'm like really close to my parents and I know that they are my everything and they have my back, and all of that package, and they spank me once in a while. I was like, yeah, I probably deserved it. I was being a little brat. I don't know. You know what I mean? So like,
Yeah.
it's just like, again, I like always want to bring it back to, it's just so much bigger than any one research paper can ever actually explain it. And the
Yeah.
media so often finds these papers and it gets really confusing.
Exactly. Yeah. The whole reason I have this podcast essentially is like to break this down and to talk about it because again, it's so individualized too. You can't just point to like one general cohort or kind of population based study. I mean, of course, when we go into the biology and the glucocorticoid receptor elements and, regulating cortisol and things like that, of course we can make, more of those generalized statements, but it really is so situational specific because of how you're interpreting things, your interactions, your relationship with the self and kind of taking that deeper dive. So thanks for bringing up that paper. I want to switch gears now to Candace and really dive into psychedelics. So that's a big part of your research. Super excited to jump in. You wrote an editorial in 2023 called What is Up with Psychedelics Anyway? And I think that's a perfect place to start. So yeah, what's up with psychedelics? It's, the new sexy hot topic that everyone's talking about. So give us an introduction there.
What is up with psychedelics? So, I think we all have like the general background narrative, right? Became popular in the 60s and like the hippies used them in Woodstock and free love and flowers. And then they became Schedule 1 substances, which means the federal government regards them as completely unsafe with no medical use whatsoever. Even MDs cannot prescribe them. High danger of addiction. So that's Schedule 1. And then a lot of propaganda came out for a lot of decades, right? So like I love asking people, what have you heard? So some of my favorites are, okay, so there's the chromosomal split. Like there was some kind of paper back in the day about like LSD is going to be like breaking open your chromosomes. And then there was the if you take LSD once, it gets stuck in your cerebral spinal fluid the rest of your life, and every time you sneeze, you're going to trip. And then of course the big, you're going to think you're an orange and peel your skin off, and you're going to think you can fly and jump out the window. So this was it. This was reiterated over and over again for decades. And so research took a sharp, stopping point. And then they've kind of come back. So there's 3 very small trials that came out of UCLA, Johns Hopkins, and New York University. I don't even know how long ago now, probably 15, 20 years ago, that looked at treating depression and anxiety symptoms in cancer patients using a psilocybin therapeutic protocol. And the results were, really, really great. Small sample size, so you can't do so much about it at that point. But it really was the jumping off point for what we're seeing now. So what we're seeing now is we have MDMA, which is the active component in ecstasy, is currently under review by the FDA for a treatment for PTSD. So phase 3 trials have been completed. The FDA will tell us their outcome in August. Psilocybin has been granted breakthrough status 2 separate times for treating depression. And we're currently still seeing a lot of phase 2 trials come out. And then there's a lot of little, and then also psilocybin for substance use disorders. We're in phase 2 trials right now as well. And I realized I gave a talk a couple weeks ago and I had a question afterwards that was like, so what happens, if they like stop taking the psilocybin and they don't feel good anymore? And I was like, oh, no. They're not like taking psilocybin. And that really made me pause to like think about that. I live in a bubble and maybe everyone doesn't understand this so much. So when we say psychedelic assisted therapy in today's modern world, there did used to be a couple different versions of it. But right now, like kind of the standard protocol that people are using is you start a relationship with your therapist. Typically, there's 2 of them. You build rapport. This is the same exact thing we do in standard therapy. And during that process, you're also choosing your intentions. You're choosing your targets that you want to be working on while the dosing session happens. That's also very similar to standard therapeutic practices today that you set goals with your therapist. And there's preparation, right? So you're told what to expect, which really helps with a lot of people's anxiety around going into a new experience that, their whole life and their parents' life were told, split their chromosomes. And so then you have your dosing session, like, and during that session, there's a lot of being invited to go inward into kind of seeing where you go yourself, but your therapists are there to support you when needed. And then after that, the different trials have different numbers, 1, 2, or 3 trials, or 1, 2, or 3 dosing sessions in the different trials, I'd say is most common. And then you have integration where you like talk about what insights you may have gained from those experiences. So that's kind of like the psychedelic model right now. We're not replacing SSRIs with psilocybin. And again, this can be really confusing, and I get why, because as the clinical trial psychedelic assisted therapy movement was growing, so was microdosing. But it's like most
It's
people don't
12:30.
realize that the microdosing movement was not scientific. It was blogs, but they all
Yeah.
rose to the same occasion. So I can see how it's very, very easy to be very confused of like microdosing's the new depression treatment. Da, da, da. So I think like more research will be done on microdosing. We will find out more. But there's very little actual like controlled, data on it at the moment.
Yeah. Yeah. And, psychedelics, we're going through this like massive change right now. I feel like in the US, they're still legal in some parts, like Oregon, I think in 2020, and Colorado in November of 2022. But really, it's going to vary, like depending on your state, the specific substance. It's been decriminalized in specific
Mhm.
cities. So
Mhm.
we know this is a huge growing interest and acceptance of these potential therapeutic benefits of psychedelics, especially for conditions that you're looking into, like depression, PTSD, and anxiety. In some cases, they may be super helpful. But yeah, so the legalities, I think, make it a little bit of a wishy-washy even subject to even talk about.
Oh
So
yeah.
yeah, they're pushing them through these trials. And then I think we'll learn a lot in August with MDMA being in phase 3.
Yeah, it's funny. My mom was visiting me a couple weeks ago, and she knows what kind of research I do, but we never really talk about it. And so she's like, "So tell me, like, it's funny, kind of like the title of my editorial. Maybe I should just send it to her. What's up with psychedelics anyway?" I think is what she said to me. She's like, "Well, like, what is it doing? Why does it work? Like, what is this?" And I was like, "Wow!" Like, and I kind of felt like I was put on the spot. And I kind of fumbled my way through explaining to her. And I was like, basically, like, this is my best, like, non-scientific way. I keep, like, this is not the way I'd say it in a paper, but I maybe say it to a person. Um, they're putting you in a state where therapy becomes much more accessible. So your cognitive capacities are not, at your highest, right? Let's just go ahead and name that. No one wants you to go driving a car when you're tripping on mushrooms because your
Mhm.
cognitive capacities are impaired. And some people might think, well, then how can you do therapy? But in reality, it kind of helps to be able to break free from the cognitive protections we have built around our narrative our whole life.
Mhm.
So like
Yeah.
whether it was trauma or whether it was just the extreme pressure to perform, a lot of us haven't actually sat in what that means to us and how it feels in our body. We avoid it, avoid, 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 avoid. So in that state, maybe like your cognitive defenses are down a little bit, and you have these profound feelings of safety and unity and love for yourself and others. So now all of a sudden, you feel safe enough in your body to go there, wherever there is for you, right? So I imagine in the beginning with these cancer patients, there was the real exploring of the fact that they're about to die.
Mm. Yeah. Yeah. I mean, that makes sense. And I've never done the psychedelic assisted therapy personally myself. I know a lot of people who have, and they explain it as something where, it's like you go to your best friend or your closest friends and you're able to give them advice, right? And tell them exactly what to do and like, feel the feelings and everything. But like you would never take your own advice or even like talk to yourself in that same kind voice or that same way. So I think
Mm.
that's an interesting and unique perspective there. So it's really, yeah, about peeling back those layers and feeling safe, which, you know, this is a fun maybe exercise too. Think of like the last time you just like sat with yourself, like you were on your phone, you didn't have some type of, I was going to say stimulus, but like stimuli
Mhm.
around you, whether it's a screen, a TV. When's the last time you actually said like, am I happy, or what am I doing today? How does that make me feel? I think we're so disconnected with ourselves even without the psychedelic therapy, that there are so many things that we can do beforehand. And personally, I just got uh I just went through a transcendental meditation program and got certified there. And, even waking up, doing, 23 minutes in the morning, 23 minutes at night, and just sitting without nothing and just having my mind run free and be able to, transcend and relax. Like, I really do think it's made a world of difference in my, anxiety per se, or kind of how I'm feeling.
Yes, I couldn't agree more. And I want to add to it that for a lot of people, they have never felt safe. Not once in their life. And that's why I'm so interested in early life trauma, early life stress, early life experiences. Because, in the first few years of our life, our brain is building. Like, it's not preset. It's preset in the ways that we are human, right? Our human brains are going to build pretty similarly. However, it is responding to our experiences. And so kind of like going back to those early examples, like maybe you weren't safe. And so your brain and your physiology kind of built up around that knowing. And so I think for a lot of these meditation traditions, spiritual traditions, and even psychotherapy traditions, which are highly similar, if anyone ever chose to sit down and actually study it all. For me personally, it was always very lonely to like be studying the spiritual traditions and be constantly referred to like, well, you can feel your inner love by, like it's like a mother's love. Ouch for all of those who never felt it. And in therapy, there's this expectation that our patients they're reaching for something, right? Like we want, like, what are your goals? Oh, to feel better. Oh, to not hate myself. Whatever. They're reaching for something that they've maybe never felt. So I think that there's this whole other level for the psychedelic experience that this is something that I can say from my personal experience. It provided me the first taste of what safety even feels like. And there was no chance that me as an individual could have reached that state without being shown what that state was.
Yeah, that makes sense.
And
absolutely.
I have an adopted son, and he's such a supernova. He's so amazing. We got him at 5. He had a lot of trauma. And I can already see the same things in him. Like I can already see that like he's 10 now and he's gotten so much love and safety and he has gotten from from his his second family all of the things, but he didn't have them the first 5 years. And I can still see how his physiology, it's stressed, it's not relaxed, and it's because it was built that way.
Yeah
So
interesting. It's interesting too how you're able to look at your son that way and really pair it up with the research and everything that you're studying. I mean, it's like an example right in front of you and someone who I'm sure you love dearly. So absolutely, I'm definitely want to go down the psychedelic assisted therapy at one point in my life. We'll see if it's within the next year, within the next 5 or so, but I'm excited when the time comes for sure.
So to get back to the biology a little bit, because I feel like
yes,
this is your epigenetics podcast and I keep taking it to all these other places.
You're good.
Which is the crux of what I do, right? Like it can be, like I am at this like molecular level, but I go out and it's fun for me and sometimes challenging because in a room of psychologists and I can speak their language and they're just like, I don't know what you're talking about with molecules. And then I try to talk to the room of biologists about molecules and they're like, I don't know what you're talking about. What is attachment? So that's the seat I sit in. Okay, so biology. We don't know what is happening at the biological level that makes the epigenome or makes these DNMTs and methyltransferases, like, why are they changing? Like, what is the signal that, the methylation on your NR3C1 gene is like, whoop, we need to change this up. We don't know. There's still so much we don't know. So what I find very interesting is the similarities between acute stress and a psychedelic state. So this is what I mean by it. So the psychological level, they're both really highly salient, right? Like if you get in a car wreck and when that memory is like in there. And for the psychedelic experience, all the older data from Johns Hopkins, it's like 97% of people said it was one of the top 5 experiences of their whole life. So you have this really salient, impactful psychological state, okay? Neurochemically, you have increased levels of serotonin, dopamine, cortisol, glutamate. Stress does the same thing. We're increasing levels of those. Physiologically, you have increased heart rate, increased blood pressure, and a couple others I might be forgetting at the physiological level, right? So when we take out the idea of, okay, well, was that traumatic or was it healing? The state is very similar. And I wonder if there's something about, and there's no data to back this up. This is me, talking about my ideas as a scientist. I'm just wondering if there's a clue there because we don't know why the epigenome responds to our experiences. I'm wondering if there's some clues because they're so similar on the outside of you can have an acute traumatic experience and your behavior changes, right? That is what we refer to as like, well, this person came back from war and now they have PTSD. What having PTSD means is behavior. So they had experiences that changed their behavior. Then if we look at the psychedelic literature and all of the non-scientific stories, people say, I had this experience and I changed. So to me, it's possible that we could use the psychedelic model to understand more about what is the biology underlying our epigenome responding to our changes in a way that shape downstream physiology and behavior.
Yeah, yeah. And so, yeah, you're seeing the same exact process if you're not assigning anything to them. You're
Yes.
seeing the same thing, essentially. So
Yes.
you have this experience, and then it's changing the behavior, it's changing the outcome. And, you notice those difference, like you mentioned, with the NR3C1 glucocorticoid receptor gene. And now you have this theory of, what can that tell us? Is there a way that you imagine actually like measuring that and diving into that? Like, I feel like it's like, yeah, how do you even go past that? How do you like study that?
Anna, the amount of ideas I have for studies, I don't have the time or money to do them all. And really even the expertise. I think a lot about cell culture being a lot of the, about being the model that potentially I'm going to need to kind of dive into these realms of my questions. I currently do not have a cell culture set up in my lab, but yeah, I think there's just still so many questions about the epigenome. So for example, in all of my research, I keep consistently coming across these OpenSea CPG sites. Do you know what these are? Should I give some background on that?
Yeah, let's give some background on
Okay.
the OpenSeas.
So in the DNA methylation world, we have really cute jargon. So we have our islands, which are CPG sites. CPG sites are just like the location in which we primarily study methyl groups, even though it's not the only place they happen. And these islands are defined by being around the promoter of a gene, which just means it's the start of the gene. And if you block the start of the gene, you're going to decrease that gene from happening. So if you look into the epigenetic literature, around exposures and psychiatric behavior and like this whole new like neuroepigenetics field, the vast majority of them are only looking at islands, like the vast majority of them, because the biological consequence of methyl groups there is pretty well understood. If you have methyl groups, you're blocking transcription, you're decreasing gene products. However, there are CPG sites across the whole gene. And there are CPG sites in between genes. And my data keep showing them be very important. But I'm looking at their association with phenotype. So there's so many steps in between what I'm looking at that I can't tell you. Like, well, what is methylation at a CPG site in the third intron doing? And I keep reading and I keep trying to read more and more and more, and I can't find a whole lot out there. So some cancer's where I go to a lot because cancer's been looking at epigenetics for a lot longer than neuroscientists have. And it's a cleaner tissue type and they get to look at their tissue. So with cancer epigenetics, they get to cut the cancer out and they get to compare it to the non-cancerous tissue, right? I rarely get to cut brains out. So it is a whole nother podcast to start talking about the peripheral epigenome and why it's related, but I would love to sometime. So anyways, so we don't know. So like there's some evidence that that they can be changing transcription levels as well, methylation across the gene. And then there's also evidence that they could be changing the isoform. So they could be changing what part of the gene is transcribed, which then down the road changes the makeup of the protein, which down the road changes the function of the protein, which is how we change behavior. And I think that's something I try to drive home for people so much of like, I'm over here talking about genes and I'm over here talking about behavior. Everything about your output, your behavior is coming from brain, and that's all proteins, baby. So
Yeah,
we talk
yeah.
about receptors and transporters and, dendritic arbiter and yeah, those are all changes in gene transcription. Those are proteins.
Yeah, yeah. It just there's so many unknowns even at the cellular level of like what are we measuring? What does this mean? Yeah, I like the cute little jargon you use there, which is yeah, so true. But is that still like a huge area of interest for you? Is really diving into the epigenetics? Like, do you think like most of the answers lie there? I mean, I know there are a lot of the entire central dogma that you need to get through to see the phenotypic outwards expression and then the behavior. So there's a lot of steps, and I understand you have to use them all together to really start to hypothesize as to why is this happening. But do you think the epigenome holds more of those answers? Like, what's your thought there?
Yes. Okay, yeah. Is your question, do I want to run a center where I have 10 different people with expertise doing the studies I want them to do? Yes. Yeah, I need people. Yeah, I need cell culture. I need more. We do animal research in our lab as well, which is really great because we get brain tissue. But even then, we're still talking about if you get a brain chunk, say I want to look at the amygdala, we're looking at different cell types. It's still very all so complicated. So yes, I want to look at the whole picture. And yes, I think the epigenome is holding a lot of information that we don't know, like a lot, a lot, a lot. I think about a lot. I
Yeah,
think about a lot about like the genetics and how much when we started the Human Genome Project, there was a lot of people that were so convinced we were about to solve all of it. And it turns out we only have more questions. Like now we know the human genome and we've learned a lot and we're more confused about the complexity of
yeah,
us. And
yeah,
I think a lot about evolution. I think a lot about like the like we all get it. We all get the idea. We've all been taught like uh a difference came out in a genome and somehow that difference was advantageous and boom, thousands of years and now more people have it. And then the other one went away. And I personally have always found that theory lacking and like, really? That's the only thing that happened. So I'm just gonna, I think that we're gonna find out a lot more about the epigenome. I think there's a lot of answers there that I think it's gonna take
yeah.
like way outside of my lifetime to answer them all.
Yeah, yeah. I use that example a lot with the Human Genome Project, and how we thought we were gonna like solve all the diseases and cure cancer, and everything would be fine. And then we're like, okay, cool, we have this information, but it's very, very limited. And like, what else is there? There has to be other reasons and other whys. So I usually compare epigenetics to the Human Genome Project. It used to cost like millions of dollars to sequence your genome. It used to take like so long, and now it's under $100. It's less than 24 hours. And I think epigenetics, like understanding your epigenome, especially being able to retest after certain experiences, which change those behaviors to start to see those differentiated methylated regions and those patterns is really what we need. And that's harder to do, obviously, in the brain because we need brain tissue. So you can just see how those complexities add up. But nonetheless, I think a really good thought experiment.
Yeah, and I will have some papers coming out soon that you'll be able to I'll follow up on me on where we
Yeah.
are looking at neuroimaging metrics, so brain structure, and looking at DNA methylation of genes highly expressed in those brain structures, but we're looking at DNA methylation for buccal cells. And we are showing that they are highly predictive in certain age brackets.
Yeah.
So it's like,
Yeah, I'm really excited to read some of those papers. So we'll definitely have to have another conversation. And I like the peripheral epigenome example you gave earlier too. So this will be the first of many. And I know we're getting to the end here as well, Candace. I want to point out I know you have the PGP or the Psychedelic Genome Project. Can you give our listeners a little bit more information on what that is, like why that's important? Can people get involved in your work?
yeah, wow. So the PGP, we want to collect samples, biological samples from clinical trials and or people using at home and or people in Oregon and soon to be Washington and Colorado that are going to clinics. And we just want a pre and a post saliva sample and then to fill out some common questionnaires after your experience. So the idea is we want to look at these baseline epigenetics and genetic predictors of how did it go for you? Did you take psilocybin? Did you have adverse events? How did you feel afterwards? And then we can kind of start to untangle, are there are there genetic profiles that we can say, hey, this is not a good psychedelic therapy for you? There's a lot of evidence that people that have similar genetic profiles, they're having a lot of adverse responses. Right now, we just exclude people from trials that if you have any family history of psychosis, you just can't be in here. And so that's an example like, well, we could be doing that in a much more modern way. And then maybe, maybe you do have a family history of psychosis, but the genetics suggests that you're not going to have an adverse event. And then we want to look at these pre-post, like you were saying before, the power of the longitudinal model with epigenetics is we can see if your psychedelic exposure is related to a change in epigenetics that explains the change in behavior that you had. So the reason, but so basically, I just described like a pretty standard idea for like a genetic and epigenetic study of exposure. However, what makes the PGP so different is because we want all the samples to come to one place. So, we are sitting on about 50, or less than that, 40 years of genetic research in psychiatry, where it was just like, it's a mess because everyone's doing a genetic study here. They're doing a genetic study here. They're doing a genetic study here. The samples get processed differently. The timelines are different. Their behavior data that they're collecting is different. And so now we need big sample sizes for genomic science. So now with the psychiatric world, we're trying to bring it all together. Like, let's everyone share our data. Let's everyone put our genetics in the same database. Let's see what we can do. And that's great. And with psychedelics, we have the opportunity to not have to reverse engineer the issue. We could just prevent it by let's all join together. Let's all psychedelic researchers around the world collect the samples and send them to the same place for processing, which is just really gold standard scientific method.
Yeah. For sure. Yeah. Well, I know I'm definitely going to get involved and look into that and submit some data, submit my samples. I'm even going to do, eventually when I do the psychedelic assisted therapy, do like my epigenome before and after on, an epic ray or something too. So I'd be super interested in looking at the NR3C1 gene on just kind of an N1 level too. So I'll get back to you on that and share some data there.
I love that you're saying that. And of course I know you're going to, but I have to, since we're, it's hard with the N of one, Hannah. I don't want to set you up for disappointment in either direction.
Yes, understood completely. So we'll
give you the group level results.
Perfect. Let's do that. We'll look at both of them.
Yeah.
Well, awesome. No, we covered so many amazing like areas in this topic. It just excites me all over again. This is exactly why I do this anyways. We're coming to the close here. One question I always ask everyone at the end, if you could be any animal in the world, what would you be and why?
Well, because life is dynamic and I'm dynamic and my epigenetics is dynamic, that has changed over the years.
I like it. So you've thought about this.
Yeah. So when I was younger, I was a bald eagle. Fierce and strong, right? Independence. Now, and then I went for a long time with a koala bear nickname because I hug with all limbs. And now we're going to go with... Hannah, does everyone always have an answer?
No. Some people struggle to answer it. Yeah. Some people are like, I know right away, but you're like going through this like evolutionary process. So I like this too. I don't think anyone has like done this.
Because you know how my mind works now.
Yes.
Okay, so my answer right now in this time of my life, I'm going to say is I believe in tools, like a toolbox. And my goal in life right now is to have all the tools, or not have all of them, is to know which ones I'm good at, and to know which ones I need to get better at, so that any given situation, I can pull out the tool that works for that situation. So with that being said, I guess I'm going to go with chameleon.
Oh, I like it. I don't know. I wasn't like expecting anything, but I was like,
I like pride myself in being authentic. And it's not that tools don't make you unauthentic. Tools, I think, make you help you be even more of who you are because you're choosing how to respond to every situation.
Right. I like it. Yeah. That's a good answer. That's a deep answer. I like it. Cool. Well, we've come to the end of this awesome podcast interview. So for listeners who want to connect with you, where can they find you? How can researchers connect with you? Can they reach out to you at the Bear Lab?
You can find the Bear Lab email at our, we are thebearlab.org. We will be pushing a very heavy recruitment effort for PGP samples by both clinical trials and individuals, whether they're going to a clinic in Oregon of their own accord or at-home users. However, we haven't started that recruitment process yet. So just keep a lookout on our website if you're interested in that.
Cool. Well, I am so excited to get this episode out. Thanks everyone for listening to us at Everything Epigenetics podcast. We'll be linking all of the research in the Bear Lab links below that Candace mentioned. And remember, you have control over your epigenetics. So tune in next time to learn more. Thanks so much.